We will see reaction of salicylaldehyde with para-amino benzoic acid in presence of acetic acid and ethanol to form yellow color Schiff base. We will see mechanism of the reaction. Nucleophilic attack by amine group of para-amino benzoic acid on electrophilic carbonyl of salicylaldehyde occurs and the enemy intermediate water is eliminated to form Schiff base. Para-amino benzoic acid, ethanol, salicylaldehyde, acetic acid. Take 1.31 gram of para-amino benzoic acid in beaker. Add ethanol to it. Boil until para-amino benzoic acid is completely dissolved. Take 1 ml salicylaldehyde. Add it to the solution of para-amino benzoic acid. Add 2-3 drops of acetic acid with constant stirring. You will observe that precipitate is forming. Orange color precipitate is formed. Then add water. Filter the product. You will see fluorescence of product in UV light. So fluorescence is seen in ethyl acetate as well as THF. Fluorescence is also observed on TLC.